Olfaction, or the sense of smell, is one of the most primal and powerful sensory systems in the human body. Unlike vision, hearing, or touch, olfaction has a direct line of communication with the brain's emotional and memory centers. The pathway from the olfactory receptors in the nose to the brain is strikingly unique. When odor molecules are inhaled, they bind to specialized receptors located in the olfactory epithelium, a small patch of tissue high inside the nasal cavity. These receptors then transmit signals to the olfactory bulb, which lies just above the nasal cavity and beneath the frontal lobe of the brain. From there, the signal travels directly to areas of the brain such as the amygdala and hippocampus, regions that are critical for processing emotion and memory. This direct neural pathway explains why smell can evoke vivid emotional reactions and detailed memories almost instantaneously. For instance, the smell of a particular perfume might immediately bring to mind a specific person or moment from the past, complete with emotional context. These reactions are not a coincidence. They are biologically encoded. Unlike other senses, which pass through a central relay station in the brain known as the thalamus, olfactory signals bypass this checkpoint entirely. This bypass allows odors to trigger raw, unfiltered responses, often without conscious awareness. From an evolutionary standpoint, this speed and directness had survival advantages. Our ancestors needed to detect danger, like the scent of a predator or spoiled food, without delay. A fast-acting olfactory system allowed for immediate behavioral responses, such as fleeing or avoiding consumption. At the same time, it enabled the recognition of beneficial smells, like those of ripe fruit or a safe environment, encouraging approach behaviors. These hardwired reactions helped shape the development of human behavior and continue to influence us today, even in highly modernized settings. The neural architecture that supports olfaction is tightly interwoven with the limbic system. The amygdala, which plays a major role in fear processing and emotional learning, receives direct input from the olfactory bulb. Similarly, the hippocampus, crucial for forming and retrieving memories, is also directly influenced by olfactory signals. This tight coupling explains why certain smells can trigger flashbacks, intense emotions, or changes in mood that seem disproportionate to the stimulus. The scent of smoke, for example, may instantly generate feelings of fear or urgency, even if there's no real threat present. The emotional and physiological response is automatic because the circuitry has evolved to prioritize potential danger. In social contexts, olfaction also serves as a subtle but powerful form of communication. Human beings constantly emit chemical cues through sweat, breath, and other body odors, signals that others can detect, often without conscious recognition. These cues can relay information about emotional state, stress levels, reproductive status, and even genetic compatibility. Studies have shown that people can distinguish between the body odor of someone who is fearful and someone who is happy, and that these odors can in turn affect the emotional state of the person who smells them. This capacity for emotional transmission through smell underscores the deep connection between olfaction and human behavior. The speed of olfactory processing also contributes to its impact on decision making. Because olfactory information is processed so quickly and tied to core emotional circuits, it can influence choices before logical reasoning has a chance to engage. People often use the phrase, "something smells off," when describing a situation that feels wrong or untrustworthy. While metaphorical, this phrase reflects a deeper biological truth. The brain integrates olfactory information as part of a larger network of intuitive processing. This system enables rapid assessments of people, environments, and objects, often tipping decisions one way or another based on subtle chemical inputs. Furthermore, olfaction plays a critical role in shaping long-term emotional patterns. Because olfactory stimuli are consistently paired with emotional experiences, they can become conditioned triggers over time. For example, a person who frequently experienced anxiety in a hospital setting may later find that the sterile smell of antiseptics triggers unease, even in a safe environment. This form of classical conditioning is made more durable by the direct involvement of the hippocampus, which helps solidify associations between smells and emotionally charged events. These associations can persist for decades, reinforcing behavioral patterns and influencing responses across a lifespan. In therapeutic settings, olfaction is increasingly being recognized as a tool for emotional regulation and healing. Aromatherapy, while sometimes dismissed as pseudoscience, has measurable effects on brain activity and mood when properly applied. Certain essential oils have been found to modulate neural activity in the limbic system, reduce cortisol levels, and promote parasympathetic activation. This ability to influence emotional states through olfactory channels highlights the potential for smell to serve as an intentional mechanism for self-regulation and recovery from stress or trauma. The rapidity of olfactory processing also makes it uniquely suited for alert systems and environmental awareness. In high-stakes settings like firefighting, chemical detection, or medical diagnostics, the ability to detect and respond to specific smells quickly can mean the difference between safety and catastrophe. Trained professionals often rely on their sense of smell as a diagnostic tool. Nurses, for example, can sometimes identify infections or metabolic conditions through characteristic odors. This underscores the continued relevance of olfaction as a high-speed, high-resolution information channel in modern life. Pheromones are chemical signals released by individuals that trigger specific behavioral or physiological responses in other members of the same species. In humans, these cues are part of a subtle but influential system of nonverbal communication that occurs largely below the level of conscious awareness. Although the study of human pheromones is still evolving, evidence suggests that these chemical signals can influence a range of social behaviors, from attraction and mating choices to mood regulation and stress responses. Pheromonal communication begins at the level of molecular detection. In many animals, specialized structures like the vomeronasal organ VNO detect pheromones. While the functionality of the VNO in adult humans is debated, research has shown that we do have receptor pathways in brain regions responsive to certain chemosignals. These signals are processed through the accessory olfactory system and interact with areas such as the hypothalamus, a brain region involved in hormonal regulation, sexual behavior, and emotional responses. The transmission of these signals bypasses cognitive filtering, activating parts of the brain associated with instinctive reactions. One of the most well-studied aspects of human pheromonal signaling involves the influence of body odors on mate selection. Studies have demonstrated that individuals can subconsciously detect genetic compatibility through scent, particularly in relation to the major histocompatibility complex MHC, a group of genes involved in immune system function. In controlled experiments, participants were more likely to prefer the scent of individuals whose MHC profiles were dissimilar to their own, potentially increasing the likelihood of producing offspring with stronger immune systems. This effect occurs without visual or verbal input, highlighting the subconscious nature of pheromonal influence. Pheromones also play a role in synchronizing behavior among individuals. One of the classic examples is menstrual synchrony, sometimes observed among women living in close proximity. While the existence and extent of this phenomenon remain controversial, early studies suggested that compounds found in sweat might subtly influence hormonal cycles. More broadly, chemical cues in human environments can affect stress levels and emotional states. For example, studies have shown that when people are exposed to the scent of sweat collected from individuals in a fearful state, their own amygdala becomes more active, and their behavior becomes more vigilant or cautious. This transmission of emotional state through scent is a powerful form of non-verbal empathy that operates through olfactory pathways rather than visual or auditory cues. The effects of pheromones are not limited to attraction or emotional contagion. They can also influence levels of trust and social bonding. Certain chemical compounds found in natural human secretions, such as androstenedione and estradiol, have been studied for their impact on mood and perception. When exposed to trace amounts of these compounds, individuals have reported subtle changes in attention, arousal, and even interpersonal evaluations. For instance, androstenedione, a compound found in male sweat, has been shown to affect women's mood and cortisol levels, even without their conscious awareness of the scent. These findings suggest that pheromones may prime individuals for certain social interactions, subtly shaping the tone and quality of interpersonal exchanges. Children and infants are also sensitive to pheromonal cues, especially in the context of bonding with caregivers. Newborns have been shown to recognize and prefer their mother's unique scent within days of birth. This early olfactory imprinting supports attachment and feeding behaviors and reinforces the emotional bond between mother and child. The mother, in turn, responds to the infant's scent, which may play a role in lactation and nurturing responses. These bidirectional chemical signals form a feedback loop that facilitates care, protection, and emotional regulation, demonstrating the foundational role of olfaction and pheromones in early human development. In group settings, pheromonal cues can contribute to the formation of social hierarchies and group cohesion. While humans do not rely on dominance pheromones in the same overt way that many animals do, subtle chemical signals may still play a role in non-verbal power dynamics. Research has explored the possibility that alpha-like traits, such as confidence, aggression, or leadership, may be accompanied by specific chemical markers that influence how others perceive and respond to an individual. This may occur through unconscious changes in body odor associated with hormonal fluctuations, particularly in testosterone and cortisol levels, which affect the composition of skin secretions and sweat. The commercial and psychological implications of pheromonal signaling have also attracted attention in fields such as marketing, fragrance design, and personal care products. While many so-called pheromone-based products on the market lack rigorous scientific validation, the concept taps into a real biological mechanism. Scents that mimic or enhance naturally occurring chemical cues may amplify social presence, influence perception, or affect mood states in ways that people are only beginning to understand. In laboratory settings, exposure to certain synthesized pheromone-like compounds has been shown to increase sociosexual behavior or ratings of attractiveness, though these effects tend to be subtle and context-dependent. Importantly, pheromonal communication operates independently of our conscious cognitive processes. People are often unaware that their behavior is being shaped by chemical cues in the environment. This subconscious influence allows pheromones to act as silent regulators of social and emotional dynamics, modulating interactions without the need for words or deliberate thought. The implications for understanding human behavior are profound, suggesting that a significant portion of our social lives is governed by invisible biochemical exchanges. Taste is one of the most evolutionarily ancient sensory systems designed to detect chemical information in the environment, particularly in food and drink. It functions as a survival-based system, enabling organisms to distinguish between substances that are safe to consume and those that are harmful. At the biological level, taste is mediated by specialized sensory cells located on the tongue and throughout the oral cavity. These cells are organized into structures known as taste buds, which are found within papillae. Each taste bud contains multiple receptor cells that respond to specific categories of taste: sweet, salty, sour, bitter, and umami. These 5 basic tastes correspond to essential nutritional needs or potential dangers. Sweetness signals the presence of sugars and carbohydrates, which are primary energy sources. Saltiness indicates the presence of sodium and other essential minerals required for fluid balance and nerve function. Sourness often reflects the acidity of a substance, which can signal spoilage or unripe fruit. Bitterness is typically associated with plant toxins or harmful alkaloids, and thus serves as a built-in warning mechanism. Umami, often described as savory or meaty, reflects the presence of amino acids like glutamate, which are building blocks of protein and necessary for tissue repair and growth. Each of these taste qualities is associated with distinct receptor mechanisms and neural pathways, ensuring rapid and accurate detection of chemical composition. The taste receptor cells transmit signals to the brain via 3 main cranial nerves: the facial nerve, glossopharyngeal nerve, and vagus nerve. These signals converge in the brainstem and then ascend to the thalamus before being relayed to the primary gustatory cortex. This region processes the raw sensory data, while additional areas such as the orbitofrontal cortex integrate taste with other sensory inputs like smell and texture. This integration gives rise to the full flavor experience, which influences both immediate reactions and long-term food preferences. The gustatory system also communicates with the limbic system, allowing taste to influence emotions, memories, and even social behavior. Beyond its sensory function, taste plays a crucial role in learning and behavioral conditioning. From infancy, humans develop preferences and aversions based on taste experiences. Positive taste experiences, such as sweetness in breast milk or a favorite comfort food, are quickly associated with reward and emotional satisfaction. Negative experiences, such as the bitterness of spoiled food or the sourness of something gone bad, can produce aversive reactions and long-lasting avoidance. These associations form early and can be reinforced through repetition, context, and emotional salience. This conditioning mechanism allows taste to become a powerful motivator for behavior, guiding choices around diet, safety, and even social rituals. The brain's reward circuitry is intimately linked with taste perception. Dopaminergic neurons in the ventral tegmental area and nucleus accumbens are activated when pleasurable tastes are experienced, creating a sense of reward and reinforcing the behavior that led to it. This mechanism underlies cravings, habitual eating, and food-related emotional responses. In situations where food is scarce or when high-calorie options are encountered, the brain amplifies the reward response, driving increased consumption. This evolutionary adaptation, once essential for survival, can now contribute to modern health challenges such as overeating and addiction to highly processed foods. Taste perception is also modulated by internal physiological states. Hunger, satiety, hormonal fluctuations, and stress can all alter how taste is experienced. For example, when the body is low on sodium, salty foods become more appealing. During periods of high energy demand, the brain may enhance the appeal of sweet or fatty foods. Hormones such as ghrelin, leptin, and insulin play a regulatory role, linking metabolic needs to taste-driven behavior. In this way, taste acts not just as a detector of external chemical cues, but as a feedback mechanism that reflects the body's internal condition and drives behavior accordingly. In addition to its nutritional and survival functions, Taste is deeply embedded in culture, identity, and social connection. Shared meals, culinary traditions, and flavor preferences contribute to the formation of community and belonging. Taste rituals often carry symbolic meaning, from religious practices involving specific foods to the bonding experiences of family dinners. These social dimensions of taste are supported by neural pathways that link gustatory perception to emotional and cognitive centers. The brain doesn't just register taste, it evaluates, remembers, and assigns meaning to it in context. Taste is also dynamic and adaptable. Neuroplasticity in the gustatory system means that taste preferences can change over time with exposure, experience, or intentional training. Children who initially reject bitter vegetables may grow to enjoy them through repeated exposure and social modeling. Adults can recalibrate their palates by reducing sugar or salt intake, allowing subtler flavors to emerge. This adaptability illustrates how taste is not fixed, but responsive to both environment and intention. The gustatory system's capacity to learn, adapt, and influence behavior underscores its importance not only for survival, but for personal development and social functioning. Chemical cues such as smell and taste, along with pheromonal signals, play a central role in modulating social bonding, attraction, and trust among humans. These cues operate at both conscious and subconscious levels, shaping interpersonal dynamics in powerful ways. The human brain is wired to detect and interpret chemical information from the environment and from other individuals, often influencing behavior without our explicit awareness. When it comes to social interactions, this sensory input acts as a form of biological communication that can trigger emotional states, modify perception, and guide decision-making processes. Olfactory signals are especially potent in creating feelings of familiarity, comfort, or caution. Each individual has a unique body odor signature that is influenced by genetic factors, diet, health, and emotional state. These scents, though subtle, carry rich information that others subconsciously detect. Studies have shown that people are more likely to trust or feel comfortable around others whose natural scent they find appealing. This reaction is not simply based on cultural conditioning, but has a biological basis, rooted in the olfactory system's direct access to the brain's limbic structures, including the amygdala and hypothalamus. These areas are critical for processing emotions, regulating arousal, and determining threat or safety in social contexts. Attraction is another domain in which chemical cues play a foundational role. While visual and auditory stimuli are often emphasized in popular discourse around attraction, olfactory cues can have a stronger and more lasting impact. The scent of a potential partner can significantly influence mate selection, even when the individuals are unaware of it. Research has shown that women tend to prefer the scent of men whose immune system genes differ from their own, potentially increasing the genetic diversity and disease resistance of potential offspring. This subconscious form of mate assessment is driven by the major histocompatibility complex MHC, a set of genes that influence body odor and immune function. In close relationships, shared chemical cues contribute to emotional bonding and intimacy. Partners often become attuned to each other's unique scent profiles, associating them with comfort, safety, and emotional warmth. The scent of a loved one can reduce cortisol levels, ease anxiety, and even improve sleep quality. This effect has been observed in experiments where individuals were exposed to the scent of their romantic partner, in the absence of visual or verbal contact. The emotional impact of these cues highlights the role of olfactory memory and emotional conditioning, where the brain forms deep associations between certain smells and positive relational experiences. Beyond romantic attraction, chemical cues also contribute to the formation of trust in broader social interactions. When individuals are exposed to certain pheromone-like substances, their perceptions of others can shift in subtle but measurable ways. For instance, compounds such as androstenedione and estrotetrenol have been linked to changes in mood, attention, and interpersonal judgment. In controlled studies, participants exposed to these compounds rated others as more trustworthy, likable, or attractive. Although these effects are modest and context-dependent, they suggest that chemical signals can serve as unconscious influencers of social evaluation and bonding. Group cohesion and team dynamics may also be influenced by shared chemical environments. In settings such as workplaces, classrooms, or athletic teams, people are constantly exposed to one another's scent signatures, which can facilitate or hinder group harmony. When individuals share similar olfactory preferences or adapt to one another's chemical presence over time, a form of sensory familiarity develops that can enhance collaboration and mutual support. This form of bonding is reinforced by neurobiological mechanisms that reward familiarity and predictability, two key components in the formation of trust and social unity. In parent-child relationships, chemical cues are especially crucial. Newborns rely heavily on scent to identify and bond with their caregivers. Within hours of birth, infants show a preference for their mother's scent, which guides behaviors such as nursing and seeking comfort. The mother, in turn, responds to her baby's unique smell, often experiencing hormonal shifts that support caregiving behavior, such as increased oxytocin release. This bidirectional chemical communication lays the foundation for attachment and emotional security, forming a critical aspect of early social development. Even in adult relationships, chemical cues continue to reinforce bonds over time. Long-term partners often develop a heightened sensitivity to each other's scents, which can act as subtle emotional regulators. The scent of a partner's clothing, skin, or hair can evoke feelings of love, nostalgia, or reassurance. In moments of separation or stress, exposure to these familiar cues can provide emotional grounding and a sense of continuity. These reactions occur not only because of past experiences, but because of the deeply embedded sensory pathways that link smell and touch with emotional memory. Social rituals and customs often reflect the importance of chemical cues in bonding and trust. From the use of perfumes and colognes to the preparation of shared meals, many human behaviors are built around the manipulation and exchange of sensory signals. The act of eating together, for example, involves a shared olfactory and gustatory experience that fosters intimacy and mutual understanding. The smells and tastes of familiar foods can create a sense of belonging and shared identity, strengthening social bonds through repeated, meaningful sensory interactions. Chemical cues, particularly those related to smell, taste, and pheromones, have a significant impact on memory, emotional responses, and decision-making. These sensory inputs are processed by the brain in a way that connects them to both conscious and subconscious processes. The interplay between chemical cues and memory is complex, involving various brain regions that are responsible for encoding, storing, and retrieving emotional experiences. When certain smells or tastes are encountered, they often evoke powerful emotional memories, often more vividly than other sensory inputs like sight or sound. This phenomenon is rooted in the neural pathways that connect the olfactory system to areas of the brain responsible for emotions, particularly the limbic system. One of the most direct connections between the olfactory system and memory is through the hippocampus, a region of the brain involved in the formation of new memories. The hippocampus is also responsible for spatial navigation and contextual memory, making it a key player in how smells are associated with specific experiences and locations. This connection explains why the scent of a particular perfume or food can instantly bring to mind vivid recollections of a person, place, or event. The olfactory bulb, which processes smells, sends signals directly to the hippocampus, bypassing the thalamus, the brain's sensory relay station. This direct pathway creates a more immediate and intense emotional reaction to smells, compared to other senses that must be processed through multiple brain areas before reaching the memory centers. Similarly, the amygdala, another structure in the limbic system, plays a crucial role in how chemical cues influence emotions. The amygdala is involved in processing emotions such as fear, pleasure, and anxiety, and it is highly sensitive to scents that are associated with either positive or negative emotional experiences. This explains why certain smells, such as the scent of a loved one's clothing or the fragrance of a comforting meal, can evoke feelings of warmth, security, or happiness. Conversely, smells associated with negative experiences, such as the odor of something rotten or the scent of a stressful environment, can trigger feelings of discomfort, anxiety, or even panic. The emotional responses to smells are often so strong that they can influence behavior in subtle ways, such as making someone more likely to approach or avoid a person, situation, or place based on its associated scent. Taste, as another chemical cue, has a similarly powerful link to memory and emotional states. The sense of taste is directly connected to the limbic system as well, particularly through interactions with the hypothalamus, which regulates basic survival functions like hunger, thirst, and arousal. When we taste food or drink, it is not just a sensory experience but also a highly emotional one. For example, the taste of a particular dish can remind someone of a family gathering, a holiday, or a time in their life when they felt particularly happy or connected to others. The taste of comfort foods, often linked with childhood or family experiences, can elicit feelings of nostalgia and warmth. These experiences are more than just about nourishment. They are deeply intertwined with the emotional and social connections associated with the food. Pheromones also play an important role in emotional responses, though often at a subconscious level. These chemical signals can affect how people perceive one another and how they respond emotionally. For example, certain pheromones can influence feelings of attraction or aversion, often before any conscious recognition of the other person occurs. In many cases, pheromones serve as an unspoken form of communication, triggering responses that are largely outside of our conscious control. This is why, for example, we might feel drawn to someone even if we cannot identify exactly what it is about them that appeals to us, or conversely, why we might feel uneasy or uncomfortable around someone without being able to pinpoint the reason. The ability of chemical cues to influence decision-making is closely related to their impact on emotions and memory. The brain's reward systems are closely tied to our emotional reactions to taste, smell, and pheromones. When we experience something pleasurable, such as tasting a delicious food or smelling a familiar and comforting scent, our brain releases dopamine, a neurotransmitter associated with pleasure and reward. This encourages us to repeat the behaviors that led to the pleasurable experience. For example, the pleasant taste of a sweet treat or the calming effect of a familiar scent can influence future decisions, making us more likely to seek out similar experiences. On the other hand, negative emotional responses to smells or tastes can lead to avoidance behaviors. The avoidance of certain foods, places, or situations is often driven by the unpleasant memories and emotional states associated with particular chemical cues. In social contexts, these chemical cues can subtly influence how we interact with others and make decisions in group settings. The sense of smell, in particular, can affect how we perceive others in terms of trustworthiness and approachability. For instance, Certain pheromones might make a person seem more attractive or more dominant, influencing social dynamics without any explicit verbal or visual cues. This biochemical influence can shape decisions about whom we form friendships with, whom we trust, or whom we decide to partner with. The chemical cues we emit, and the ones we detect from others, contribute to the invisible yet powerful forces that drive social interactions. In conclusion, chemical cues such as smell, taste, and pheromones are intricately linked to memory, emotional responses, and decision-making. These cues can influence behavior in profound ways, shaping our perceptions, social interactions, and personal choices. Through the direct connection between sensory input and brain regions involved in memory and emotion, chemical cues have the ability to evoke strong emotional reactions, trigger powerful memories, and guide our decisions without our conscious awareness. These subtle but potent influences remind us of how deeply our sensory experiences are intertwined with our emotional lives and the decisions we make. Chemical cues, including smell, taste, and pheromones, are not only critical for survival and emotional regulation, but also serve a powerful role in shaping human behavior, preferences, and interactions in complex social environments. These cues affect everything from individual food choices to the dynamics of social bonding, emotional regulation, and even the formation of long-term relationships. The impact of these chemical signals goes far beyond simple physiological reactions, as they intertwine with our emotional and cognitive processing to guide behavior in often subtle and unconscious ways. At the most basic level, smell is one of the most primal senses in humans, with deep evolutionary roots. The human olfactory system is directly linked to the brain's limbic system, which is involved in processing emotions and memories. This connection explains why a specific scent can trigger vivid memories and emotional reactions. For instance, the smell of a particular flower, a dish, or even the scent of a place can evoke intense feelings of nostalgia, happiness, or even sadness, often without us consciously recognizing the source of the emotional response. The deep connection between scent and emotional memory can be traced back to the survival advantages it provided in evolutionary history. The ability to recognize familiar smells could alert early humans to the presence of danger or help them identify safe and nourishing food sources, thus influencing both immediate survival behaviors and long-term emotional responses. This link between scent and emotion plays a crucial role in the social behavior of individuals. People's perceptions of each other can be influenced by the subtle, unconscious processing of smells. The body emits a variety of scents that are not only influenced by external factors such as hygiene and environment, but also by internal biological processes, including genetics, hormones, and health status. These natural odors can influence how people perceive one another in terms of attractiveness, trustworthiness, and even overall likability. For example, research has shown that women tend to be more attracted to the scent of men with immune system markers that are genetically different from their own. This unconscious attraction is thought to have evolutionary benefits, as it increases genetic diversity and the likelihood of producing offspring with stronger immune systems. This dynamic demonstrates how deeply ingrained our olfactory systems are in our understanding of social interactions and partner selection. In addition to the personal and emotional effects, chemical cues also serve as an invisible form of communication in social settings. Pheromones, which are chemicals that carry information between individuals of the same species, can subtly influence social behaviors such as dominance, mating, and cooperation. Humans, like many animals, produce and respond to pheromones that can affect a range of social dynamics. Though human pheromones are not as well understood as those in other animals, Studies suggest that they play a role in regulating sexual attraction, social cohesion, and emotional responses. These chemical signals can affect moods, perceptions of others, and even influence decision-making without individuals being consciously aware of their presence. The ability of pheromones to subtly shape how we interact with others speaks to the power of chemical communication in regulating complex social behaviors. Taste, similarly, holds profound implications for human behavior, particularly in relation to food preferences, emotional regulation, and social interactions. From an evolutionary perspective, taste serves as a mechanism for assessing the nutritional value and safety of food. Sweetness generally signals energy, dense foods that are safe and beneficial, while bitterness is often an indicator of toxins or spoiled food. As a result, taste preferences are not just learned, they are shaped by biological imperatives that guide survival behavior. The emotional and sensory pleasure derived from certain tastes, such as sweet foods, is tied to the brain's reward centers, releasing neurotransmitters like dopamine, which reinforce behaviors linked to eating and consumption. This connection between taste and reward is deeply embedded in the brain's architecture and helps to explain why we may develop cravings for certain foods or why some foods are particularly comforting. Comfort foods, often associated with childhood or family traditions, can trigger positive emotional responses, providing not just sustenance but emotional satisfaction as well. The link between taste and memory is also notable, as the taste of certain foods can evoke powerful emotional responses tied to past experiences. For example, tasting a specific dish that was frequently prepared by a loved one can bring back memories of family gatherings, childhood, or a particular period in life. This deep connection between taste and emotion is not just a result of personal experiences, but is also influenced by cultural factors. Shared food traditions within families or communities help strengthen social bonds and create a sense of identity. The act of eating together, sharing a meal, and engaging in communal food practices can deepen relationships, strengthen group cohesion, and reinforce cultural heritage. Another key aspect of how chemical cues shape human behavior is through their impact on decision-making. Both smell and taste influence our daily decisions, from food choices to social interactions. Chemical cues often affect our subconscious judgments, guiding us toward or away from certain experiences based on emotional and sensory cues. For instance, the smell of freshly baked bread or a favorite dish can evoke a sense of comfort or pleasure, motivating us to seek out those experiences more often. On the other hand, unpleasant odors or tastes can lead to avoidance behavior, steering us away from certain situations, people, or places. The influence of these chemical signals on decision-making underscores their importance in shaping our everyday behavior, even when we are not consciously aware of it. In social contexts, the role of chemical cues extends beyond individual preferences and behaviors to broader group dynamics. Smell, taste, and pheromones contribute to the formation of trust, attachment, and cooperation in groups. Humans are social creatures, and our interactions with others are deeply influenced by the subtle cues we receive from those around us. These cues can impact everything from romantic attraction to professional relationships, helping to shape how we perceive and connect with others. The power of chemical cues in social behavior highlights their critical role in human interactions, influencing everything from individual choices to the larger dynamics of groups and communities.